So the drama. That's the drama. The drama is cancelled. And the question is drug discovery really a drama? And we say, no, it's not. And it's because ChemAxon rocks, as you could hear yesterday evening, but it does not rocks. If you know what I mean. If you don't, I hope that by the end of my presentation, you'll find out what I mean. But regarding the drama, why it's cancelled, the idea was that 5 of us who works in this drug discovery tools group, as Metzi kindly introduced us, would do a kind of role-playing, a real drama, as if we were medicinal chemists and modelers in a pharmaceutical company or a small biotech group, and each of us play a certain role, and we try to do, well, a very narrow, limited part of the long drug discovery process, the virtual screening, and perhaps some profiling using ChemAxon tools. But as we thought about this, it turned out that not many of us would feel comfortable in role-playing. So we decided to do just a plain and boring presentation instead of the drama. So that's my first real slide. Do you remember this slide? You should have seen it. Yes, correct. That's Alex Driver's slide from the last UGM. And do you remember what he said with this thingy? He used a very nice analogy. Can you recall? Right. Exactly, yeah, Shimano. Shimano is a company that makes parts for bicycles, all kinds of accessories and parts. And he used this analogy that ChemAxon is kind of the Shimano of ChemInformatics, because we also make components, parts, that can be easily integrated into many third-party tools or pharmaceutical discovery systems, just as smoothly as you can fit a Shimano brake on your bicycle, whatever make your bicycle is. And I really like this analogy. And I started thinking, if our group, the Discovery Tools group, does really produce these kind of tools, components, Shimano-like ChemInformatics discovery tools that can be smoothly integrated into various platforms, tools, applications? And the answer is yes. The screening tool that we plan to use in the drama anyway is such a versatile component that, that can be integrated and actually is integrated in various platforms and tools. Now, what's screen? Screen is a tool, Screen is a tool to perform high-throughput virtual ligand-based screening. It's very important. Of compound libraries using various similarity comparisons by various molecular descriptors. That's a list of some of the available descriptors. And as I said, the tool is quite component-like and is already integrated into many applications. It's just a brief list. JChem for Excel. Tamás gave a very nice demo a couple of hours ago how smoothly screen is integrated in JChem for Excel. He did a pairwise comparison and generated a heat map by the Similarity scores. I really like that. And screen is also available in instant JChem, of course in JChem base, in the cartridge, accessible via JChem web services, is integrated in KNIME, Pipeline Pilot, and InfoSense workflow platform, but is also available as a standalone command line application programs. It's just to entertain myself. That's not for users, really. I skipped the JChem for Excel demo because I enjoyed Tamashi's demonstration very much, and I think you could get an impression how easy to use JChem for Excel for similarity searches. That's instant JChem. That's a default table. If you have instant JChem, then you can get these default structures, and just go to query, draw a query, or I learned something from Ákos on the first day of UGM. That's the fastest way of drawing structure for non-chemists like me is just import them by name. Oops, excuse me. Okay, so aspirin will be our query. And here I can change the search mode from substructure, which is Szabolcsi's business, and he talked about it today, to similarity, and use this aspirin as a query structure with a similarity threshold of 0.5. That's quite safe. I mean, we would get something. And just run the query and browse the structures. So these are the structures in this default database that have a similarity less than 0.5. This similarity less than 0.5. So it's so smooth. Back to the presentation. That's a KNIME workflow. Timmy demonstrated how easy to use KNIME is. I would like to show how you can use KNIME for similarity searching. So just go to the ChemAxon nodes, the JChem extension, and if you would like to perform a similarity calculation, then among manipulators you find more similarity. Just drop in that node. And then all you need to do is to ensure that you have some structures to search for. This should be the target structures and the query. So we will enter the query by the Marvin sketch, and we will import, let's say, from an SD file or from a SMICE file, the target structures. And at the end, we will visualize the hits, the virtual hits. So the workflow is very easy. And to configure it is very straightforward. We configure the more important node to specify the input. It's not KNIME, which is so slow, and not the JChem nodes, but my laptop. It's not a complaint. I don't want a new laptop. I like this. Okay, so more important is configured. Then we configure Marvin sketch. So the query, we just draw the query. It's much faster when I entertain myself. So aspirin again. Okay, and it's done. And just execute this workflow. So as you see, the more importer started to read the input SMICE, and when done, the more similarity starts running, and now the results, the hits, are ready for viewing. So if we just peek into this node, we get a nice table of the hits. That's it, along with the structures, along with the similarity scores, well, the similarity scores in this case. So KNIME is the third example in which similarity calculation is integrated, and you can use it really easily and smoothly. I skipped this one. I really like it. because as you could see, my Safari browser just crashed when I started my presentation. But if you go to the if you go to the ChemAxon website, then you will find this nice Ajax implementation of similarity searching and substructure searching. So here, if you specify a query, you can run a similarity search, and you would get the hits, again, along with the similarity scores. I will skip this demo as well. It's, for me, you can use JChem-based to perform similarity searching. You can use the JChem manager to import structures to your database. Then you can modify the database table and add molecular descriptors. And then you can go command line is another alternative to perform similarity searching. You can go command line and use these commands, and the obtained results can be, for instance, visualized by Marvin view, and you get this nice table view. Okay, so we saw quite a few examples of this Shimano-like behavior of similarity searching. But the question is, the big question is, if we consider that we are in the drug discovery drama, is this useful? Does it work? Well, yes, but it's not that straightforward. That's one reason why drug discovery is hard. If consider this situation, these two green compounds, or greenish compounds, are both actives on the same target receptor. This thing is not. Yet, if you use similarity calculations and you compare these three against each other in a pairwise manner, you find that this non-active compound is more similar to either actives than the two actives to each other. And if that's the situation, then you cannot really expect that your similarity searching will give you tangible, good, reliable results. So we need to do something to improve the basic similarity searching and similarity scoring. And what we do is we take the regular similarity metrics and we optimize them in a way to make them perform better on a given but arbitrary active family. And by this optimization, we can achieve a much better similarity score between the two actives than between the non-active or inactive compound and two actives. And if we manage to fine-tune Tanimoto or other similarity score, which is used in virtual screening, to behave like that, namely be more sensitive to actives, then we can expect good results. So if you carry out this optimization, which is very straightforward, is absolutely automated, you just run one command and you get the optimized and fine-tuned similarity metrics. What this engine does inside and under the hood is we take the standard metrics, introduce some new tunable free parameters, and in this automated tuning procedure, we make the metric more selective against the actives. And by this, we can achieve very nice enrichment. That's, which one? This bluish curve is the ideal, is the best, is when the hits from a virtual screening exercise have the high similarity score. And you can't go below this line. But the better you come close or approximate this line, the better your virtual screening performs. So that's what the ordinary Euclidean metric does. That's what the ordinary common Tanimoto metric does. And with this optimization procedure, we can very well achieve the theoretically best performance in virtual screening. That means that we will get good hits in a virtual screening. But how do you validate your hits? Okay, we see some enrichment curves and things like that, but are the hits that we get from virtual screening good ones? We take an example, this Rho kinase receptor, and we played with this. That would be the drama. So I just show some snapshot of this exercise. We ran virtual screening using an optimized metric. And, excuse me, there are a few hits along with the similarity scores. In this exercise, we use the pharmacophore fingerprint. That's just one descriptor available in the screening tool. And then it took these hits, virtual hits, and tried to see how good they are. And the exercise what we did was we aligned these virtual hits on the 3D structure of a known active compound. Okay, so we knew that there was an active compound, I mean active in this receptor, the Rho kinase. We knew it from an HTS. And we took the 3D conformation of this active and overlaid our virtual hits on top of this molecular structure. And as you can see, the overlays for these 16 compounds are very nice. Okay, they have different molecular scaffolds, different molecular structure, yet they can take very similar molecular geometry. And that's essential, that's needed to make sure that these virtual hits would fit into the receptor, into the binding pocket of the enzyme. So how does this alignment work? What's under the hood will not be told here, but I just would like to give you an impression. So alignment means that we take two molecular structures or more. These can be either 2D or 3D structures. And in an exhaustive search process, we try to find the 3D conformation of these two compounds that match in the space. How do we do that? We take the compounds, but instead of performing an atom pairwise based overlay of these two structures, we calculate an approximate volume for each compound. And during the overlay, we use these molecular volumes. And the goal of this overlay procedure is to maximize the overlap, or better said, the intersection of these two molecular volumes. And we say that the two compounds are overlaid or aligned when we find the maximum intersection of the two molecular volumes. Now, using this alignment tool, we can go one step further, and instead of taking an active compound out from the binding pocket of the target receptor and do the overlay on top of it, as we saw in the previous slide, we can actually do this overlay exercise or alignment just inside the binding pocket of the receptor. And here's a brief animation using Marvin space, how we achieve that. So that's the protein. And there in the binding pocket, you see a natural ligand. And this ligand was not used during optimization or searching. And we take another structure, for instance, one which was found as a virtual hit. There it is. So you see, is quite apart from the cavity. it has arbitrary coordinates and arbitrary conformation. It can be a 2D structure, and actually in this exercise, it's taken as a 2D structure when it's overlaid on the natural ligand. So that's the structure to be, well, we sometimes say to be docked inside the binding pocket. But for this docking, pseudo-docking, I should say, we don't use the receptor, but we use the natural ligand. So we shouldn't call it docking. And as you see, that's all real time. So now these are the two structures, the original ligand, natural ligand, and that will be overlaid on it. And that was real time. We found the binding conformation, we can say, of this compound. This one, that one. Okay, so we can say that if we manage to overlay this virtual hit on the natural ligand, then we can expect this being an active compound. So that's another way to validate a virtual screening. Alignment, which I said is flexible. That means it can take either 2D or 3D conformations for the input compounds and can tweak the two or more structures on top of each other by maximizing their volume intersection. Has been developed recently, and it appears to be a very generic engine. And we managed to find some further applications of this alignment engine. One is the prediction or calculation of the molecular volume. In this case, we don't have to deal with two molecules, just one. So it's hard to see why alignment can be used and is actually used in this exercise. But it's not alignment, but the underlying calculations which are used in this exercise. We can predict the molecular volume of arbitrary structures to very high accuracy. And again, using the same machinery, we can predict what we call the minimum projection area of small molecules. This is related to the minimum intersection of the 3D conformation of the molecules. And besides of the minimum projection area or minimum intersection, we can also calculate the maximum size or length perpendicular to the plane of this minimum projection of the structure. So we can figure out how thin this molecule can be, and when it's the thinnest, how tall or how long it is. And these two properties are very closely correlated to various membrane permeability properties that we do not want to predict yet, but we have some partners in the academia who use these kind of calculations to predict such molecular properties to very high accuracy. So what's in the pipeline and what next? We will release a 3D simulated searching based on this alignment motor or alignment engine in the next JChem release, along with various shape descriptors, 3D descriptors that can be calculated using, again, this alignment tool. We have various ideas. And what we try to achieve is basically 3D virtual high throughput screening, something like OpenEye Rocks does, perhaps faster, perhaps to higher accuracy, but using this flexible alignment technology. As I said, this alignment machinery is quite general. So we believe that we can use this to more traditional 3D pharmacophore searching, which unlike ROCS takes a 3D pharmacophore model and during the screening, it takes the target structures and fits them to this pharmacophore model instead of to a query structure. What we also learned is not just that customers are interested in 3D descriptors and 3D screening, but also that the real value in virtual screening are the molecular descriptors themselves. So what we do now, we extend our existing set of molecular descriptors. Is already fairly comprehensive, but for instance, on user demand, I'm sure you won't be surprised, we add the pipeline pilot extended connectivity fingerprint to this screening tool, along with the 3D shape descriptors, and these should come out in the next major release of JChem. And finally, I would like to thank my colleagues, the members of the discovery group, their work, in particular Adrian, who's the brain behind this alignment tool. He worked not only on the implementation, but on the concepts. He did all the heavy lifting, and I thank you, Adrian. It involved lots of math and thinking and 2 years' work, and now we feel confident that with the current engine we achieved our goals in terms of speed and accuracy as well. Ödön and Gábor contributed to this work, I should say, in the past 10 years. Ödön is the brain behind the state-of-the-art optimizer, which is used in various 3D tools at ChemAxon, including the alignment tool, but also in 3D structure generation that you could see on a poster presented yesterday. Gábor is an endless pool of ideas, I should say. So he helps other members whenever is needed. And Timmy provided us with the raw kinase actives and helped us understand this tiny portion of the drug discovery process that we call virtual screening better, to help us see how these tools fit in a real, not industrial, but real virtual screening exercise. Zsuzsa implemented the optimizer for the metrics. So when we achieve the best metric, the best performance and the highest enrichment in virtual screening. And I should mention Judith, who worked on Marvin Space, which is a 3D visualizer, but gradually becoming more. It won't ever be MOI, but we incorporate these more like modeling tools in Marvin Space, because Marvin Space provides a quite natural framework for this more modeling-like applications. So that's why I put her on the slide, and I thank their work here. Thank you for your attention, and I'm happy to answer your questions.
Okay, so we'll go start questions here.
Just a quick question. So you're, generally, the OpenEye software first works by you enumerate a conformational library from a single molecule and then do a ROC screen on that conformational database, and they have a new new sort of compound descriptor as a text file which is multi-conformational, so it's very, very fast. Do you feel that you're able to include some conformational searching in your volume fitting, and how would you make it fast so it can do thousands of molecules a second?
Yes, early results are quite encouraging, so we believe that both in terms of speed and accuracy, we can achieve quite similar performance to ROCs without the need of multiple discrete 3D conformers. we may need, and I'm sure we will use, some kind of pre-filtering based on 3D descriptors. But what's important that these 3D descriptors will be calculated again using this alignment tool.
Well, may sound like philosophy, but in fact it is not at all. You know, there's an issue when you show the similarity slides. You said these two molecules are active, so they have to be similar. Beware, this is wrong. You know, the similarity principle is skewed and one-sided.
You're right. Yes.
Yeah, and all Al-Qaeda members are Muslims, but not all Muslims are Al-Qaeda members.
That's correct.
And, if the day you will have two binders which go to the same site but use different anchoring points and different sub-pockets, which are as different as they can be, you could still force them by metric fitting to give a small score, but then strange things will happen in your descriptor space.
Yes, but what I have not mentioned, we can add, and we've been working on, kind of colored volumes in which we go beyond the pure and bare geometry, and we color the surface and the volume by whatever property, can be pharmacophore properties or extended atom types. And during this fitting or overlay, we can maximize their intersection.
Yeah, well, if there is a set of descriptors that make two active molecules naturally look similar, it means that they are similar. That it's just that the similarity was hidden. But they could also be genuinely different, and there should be no possible descriptor space in which they would look alike. You have to take care of these tricky issues.
Okay.